So the restriction enzymes are the exonuclease and endonuclease. These are the large class of enzymes. These are the nucleases. These are two kinds, exonuclease, endonuclease, that is within the specific position within the DNA. Endonuclease and exonuclease from the ends. It removes the nucleotides and then it attacks on the palindromic nucleotide sequences. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA. Each restriction endonuclease. And it says restriction enzymes cut the strand of DNA a little away from the center of the palindromic sites, but between the same two bases on the opposite strand. This leaves a single stranded portions, that is the work as the sticky ends, and because they form the hydrogen bonds with their complementary counterparts. This works as a DNA ligase. The restriction enzyme cut the strand of DNA a little away from the center of the palindromic sites, but between the same two bases on the opposite strands. And it says each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA. It's saying about and EcoRI restriction endonuclease enzyme EcoRI. Again, it says that each restriction endonuclease functions by inspecting the length of a DNA sequence. Once it finds a specific recognition sequence, it will bind to the DNA and cut the each of the two strands of double helix at specific points in their sugar phosphate backbones. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA. Each restriction endonuclease functions by inspecting the length of a DNA sequence to the DNA and cut each of the two strands of the double helix at specific points in their sugar phosphate backbone. So these are the characteristics of restriction endonuclease. What about the exonuclease? It removes nucleotides from the ends. It's given in here. Anyways, but restriction enzyme cuts a little away from the center. Restriction enzymes cut